Hello and welcome to the week 9 question and answer session of the NPTEL course Experimental Biochemistry. We will be beginning in 5 minutes from now. Let the others join and then we can begin. So in today's in today's discussion, we will be talking about the isolation and characterization of nucleic acids. Just like we did for proteins a few weeks back, we are going to do the same thing for nucleic acids this time, and we are going to know how nucleic acids are isolated from different natural sources, from bacterial sources. Okay, and then how exactly we purify them, because inside the cell there are so many different compounds. There are proteins, there are polysaccharides, and there are so many different other biomolecules associated with these nucleic acids. And then how do we remove all those impurities and get a very pure fraction of nucleic acids. Then also we will learn why exactly this is so important. Why do we need nucleic acids? Okay, so what all applications do pure nucleic acids actually have? So we will be talking about all that today, and then we will learn about how to find out the purity of nucleic acids. Okay, so finally we will be solving a lot of MCQ questions, MSQs. Okay, and please stay tuned, and we will be starting in 5 minutes. So we are back and let us begin with today's talk. Okay. All right. So welcome back to the session. This is coordinated by Professor Sharita Dasgupta and Professor Shomode from IIT Kharagpur, and I am Bias Mukherjee, a research scholar in Dr. Shomode's lab, and today I will be discussing with you the topics of isolation and characterization of nucleic acid. Okay, so I have already given a very brief outline of what is there in today's discussion, so let us carry on. Okay, so firstly, let us talk about nucleic acids. So in the last few videos, I am sure you have seen, if you have followed my videos, I am sure you might have understood by now that there are 4 different kinds of biomolecules inside the cell. They are nucleic acids, proteins, carbohydrates and lipids. Okay, so nucleic acids, proteins and carbohydrates, these are biomacromolecules, and the lipids are very small in size and therefore they are micromolecules. Okay, now nucleic acids can be divided into 2 different types. They are DNA and RNA. Okay, and what is the difference between DNA and RNA? DNA is called deoxyribonucleotide. One second. Yes. Okay, so DNA is called deoxyribonucleic acid and RNA is called ribonucleic acid. Now, why is this name given? If you see the structure of DNA, okay, if you see the structure of DNA, you will see that if we count this way, okay, so if we count and clockwise, if we, one second, yeah. Okay, so if we count clockwise, this is carbon number 1, this is carbon number 2. In the second carbon, instead of OH, there is a single hydrogen in DNA. This makes DNA less reactive. Okay, and that is why DNA is more stable compared to RNA. We will look into that very soon. So this is basically made up of this phosphodiester backbone, and this phosphodiester backbone is made of phosphate group and ribose sugar. So this is the ribose sugar. It is a this is the sugar. Okay, so there are 5 different carbons in it. This is the ribose sugar, and this is the this is the nitrogenous base. So there are 4 different types of nitrogenous bases. You can see in DNA, these are the 4 different types of nitrogenous bases in DNA, and in place of thymine, there is uracil in RNA. Okay, so now if you see in DNA, what happens, the 2 strands, they are wrapped in a helical fashion around one another, and these 2 strands are joined to one another by the hydrogen bonds between the nucleotide bases. Okay, so these are the nitrogenous bases, and between the nitrogenous bases, between A and T, there are 2 hydrogen bonds, between G and C there are 3 hydrogen bonds, and that is how the 2 strands of DNA go around one another, forming a double helix, and they are joined. So what happens, the bases basically they are stacked together. The bases are having this ring structure, right? So they are stacked upon one another, and they are very well protected from the outside environment. But in case of RNA, what happens, that it is single stranded, and so the bases are more exposed to the surrounding, and therefore they are more exposed to the environmental condition. That makes RNA less stable. That is point number 1, and the second point is its OH group in the second position. That makes it more reactive. Okay, so DNA is therefore the storehouse of genetic information, while RNA participates in the transfer of this genetic information to proteins for performing various life processes. Okay, so if you take only the base and the sugar, this is called nucleoside, but when phosphate group is added to a nucleoside, it makes up a nucleotide. Okay, these are small conceptual questions. I'm sure you remember this from your 10th and 12th class. Okay, and these are the different bases. You know what is purine and what is pyrimidine. I'm sure everyone knows. So we will move on. Okay, so what about this RNA that makes it so susceptible to degradation? So if you look at this structure, this is actually the structure of RNA, which is having this 2' OH, and it is also having this 3' phosphate group attached, right? So it's a different strand. It's one single strand made up of different nucleotides. So what happens? Let's say there is some higher pH, so alkaline condition. What happens is there is OH in excess under alkaline condition. So what it does is there is a nucleophilic attack on this 2' OH, because of which there is a cyclic uh, derivative form. This is called the 2', 3' cyclic monophosphate derivative. So what happens, because of this nucleophilic attack, this oxygen makes a bond with this phosphate, and that is why this bond is opened. The bond with the next nucleotide, it is opened, and therefore the RNA gets cleaved, right? That happens. And again there is in presence of water, this mixture of 2' and 3' monophosphate derivatives are formed. So it is no more a cyclic product, but it is a shortened RNA. See, this is the derivative. This is how the derivative looks like. So this is how RNA basically gets chopped off in higher pH. Again, there are different enzymes. They are called the RNases or the ribonucleases. They can catalyze RNA hydrolysis. They are basically chopping of RNA. Then there is heat denaturation. So what happens in heat denaturation? See, RNA, the bases are very much exposed to the surroundings, right? And because of that, RNA can get degraded when heated at 65 to 70 degrees centigrade for 15 minutes, faster at higher temperature. And therefore, it is always stored as at minus 80 degrees centigrade to make it stable. But then the thing is that for DNA, I am sure you remember that for PCR reactions, we heat it to a temperature of 95 degrees centigrade. Only then the denaturation of the two strands happen, and then the rest of the reaction can proceed. So DNA does not get degraded there also, but RNA gets degraded very easily. Okay, this is why working with RNA is very difficult, right? So in our lab we work with RNA, and we need to be extra cautious with everything. So look here, RNA is susceptible to degradation, and you can see here, right? So you can see here that it is susceptible to degradation. This structure of this enzyme that you see towards your right, this is basically RNase A, and RNase A has a property. I am sure you remember studying about RNase A in a previous lecture. So RNase A has this property of hydrolyzing or breaking single-stranded RNA. Okay, so RNases are released from the compartmentalized condition in cells upon lysis. So basically, when we will study about this isolation and purification of nucleic acids. So what is isolation? Isolation means separation from all other different compartments, all other different components, right? So separation of the nucleic acid from every other biomolecule, that is called isolation. What is purification? Making sure that there is no impurity of any sort in that solution, nucleic acid solution, be it DNA, be it RNA. There is a homogenized form, only a single particular form is present there, that is called purification. So that is your isolation and purification. Okay, so now we will see this. So RNase is basically what happens to get DNA or RNA from the cell. You need to break open the cell, and when you break open the cell, you know that there are different compartments in the cell having different pH. Inside those different compartments, there are different enzymes which will be active only at specific pH. And then what happens when the cell wall is ruptured, and inside the cell the different organelles, they are also ruptured, then what happens? All these enzymes come up, they come out, and then they may be active. So there are RNases inside the cell also, right? So they will be active. And then to make sure that they are not cleaving the RNA of interest, we need to add different RNase inhibitors, which are going to go and inhibit these RNases. Okay, and this is the process of lysis. So lysis basically means breaking open. Okay, so they are ubiquitously present on the skin. So these RNases that we are talking about, they are present everywhere. So it is like, as we have studied about bacteria, they are ubiquitous, they are present everywhere. Similarly, RNases is also uh, ubiquitous. So when you heat RNase, it is a very stable protein. It is not going to get denatured. It is going to be very active, and therefore you need to doubly autoclave water. So when you autoclave water 2 times, then what happens? There is a heat of 121 degrees centigrade for a prolonged duration of around 2 hours. Then maybe RNase gets destroyed. Other than that, for only once, if you autoclave, it is not going to get destroyed. Okay. Besides that, RNase does not require this EDTA, it does not require any metals for its activity, right? So unlike DNase, RNase does not require any uh, divalent cation, and therefore even if you add EDTA has nothing to chelate there, and it cannot stop the activity of RNase. So look here, there is no simple method to inactivate RNases because it contains a number of intra-chain disulfide linkages. So RNase has disulfide linkages that makes it extremely stable, and it is very well folded and resistant to the inactivation by prolonged boiling and mild detergent. It gets quickly refolded after denaturation. So also unlike DNAs, RNAs do not require divalent cation for their activity and therefore cannot be inactivated by the inclusion of EDT or other chelators. Okay, and therefore the best way to prevent problems with RNAs is to prevent contamination. And therefore when you are working with RNA, you need to have a completely separate setup for RNA. So there has to be a different set of pipettes, different working area, preferably different tick boxes, everything. Okay, so everything that you are going to introduce into your RNA, it needs to be separate from everything else that you are using for any other different procedure. Okay, now let us look at the different applications. So it's given in a very beautiful colored format so that you can remember. So see, pure DNA, it can be used for genetic engineering, it can be used for forensic science, forensic studies, molecular diagnostics, therapeutics, whole genome sequencing, cloning. So all of these, what exactly are these things that you can find out, you can Google. I'm sure you would be interested. So please learn more about all of these things. You will get more interest in the subject. Okay, and pure RNA can be used for gene editing, vaccine preparation, therapeutics, and IVT, that is in vitro transcription. There is protein replacement therapy, molecular diagnostics, cDNA library, and so on. Okay, I would recommend you to read about all of these different topics. That is going to give you better idea and make you more interested in this subject. Okay, now, so this tells us that there are so many different applications of pure DNA and pure RNA. So that tells us how important it is for us to purify DNA and RNA. Fine. Now, how do we purify DNA and RNA? So people have come up with so many different methods, and this has been going on. Isolation of DNA, isolation of RNA. If you read the history of these procedures, you would come to know that people have been doing this since a very long time. Okay, so they have been using different kinds of methods, and we will learn about them now. So there is phenol chloroform separation. This is also called the organic extraction method, in which what happens, they utilize phenol and chloroform to separate the nucleic acid from proteins and other contaminants. It involves multiple phases of extraction and careful handling of hazardous chemicals. So we will look at phenol chloroform method of separation very soon. Then there is silica based columns. So this is used now, okay, in laboratories. So what happens, there is a spin column. It is like a small column. Inside that column there is silica beads. Okay, to the silica, when you add this solution containing your nucleic acid, the solution will contain some salts in it. Let's say there is sodium. Okay, and this is a chaotropic agent. So chaotropic agent goes and binds to the silica. Silica is SiO2, right? So it is negatively charged. Salt is positively charged. So it goes and binds to silica, and then what happens again, salt is positive, and so when your DNA comes, it binds to the salt. So salt basically forms a bridge between the silica beads and DNA. Okay, so then what happens, the DNA comes and binds, and all other unwanted impurities, they flow through, then you elute it with a specific buffer. Okay, so let's look at this. Using silica membranes or beads to selectively bind nucleic acid while contaminants are washed away. After binding, the nucleic acids are eluted from the column using low salt buffer or water. Okay, so popular kits include spin columns and 96 well plates. Then comes in guanidinium isothiocyanate or GITC. This is mostly used for RNA isolation. So GITC, what it does is it denatures protein and solubilizes nucleic acid. Then after that, ethanol or isopropanol is added. This is used to isolate nucleic acid from any other contaminants, and this GITC is commonly used in trizol. So trizol is a ready-made reagent for RNA extraction. We will learn about trizol in the next few slides. Okay, then there is magnetic beads. So what happens here is that certain magnetic beads coated with molecules that bind to nucleic acid, they are added in, and then what happens, these nucleic acid bind to the beads, and then they are selectively captured using a magnet. After washing, the nucleic acids are eluted for downstream application. Then there is Chelix resin. So this is new. So what happens here is, see, Chelix resin binds to metal ions present in the sample, preventing them from interfering with the downstream applications. After binding, nucleic acids are released by heating the resin, and contaminants are removed by centrifugation. So you can learn about this, about Chelix resins. We do not have questions on this actually, and so we will not be looking at this in very high details. Okay, now comes in other methods of nucleic acid purification. So there is anion exchange chromatography. Now in anion exchange chromatography, what happens? This relies mainly on the principle of ion exchange to separate nucleic acids from contaminants based on their charge. Now again, you can understand this cannot differentiate between your RNA and DNA. Of course, both are negatively charged. So nucleic acid bind to the resin under specific pH and ionic conditions and are subsequently eluted using a salt gradient. Fine. Then there is organic solvent precipitation. Here what happens? This involves addition of ethanol or isopropanol. I told you, right? And then what happens? The nucleic acid basically it precipitates out. Okay. So how does it precipitate out? Your ethanol is interacting with water, and the nucleic acid is no more able to interact with water. So the interaction between the solute and solute, that is nucleic acid is so high compared to the solute solvent interaction that there is precipitation. It basically interacts with one another and then precipitates out, right? Then what happens after centrifugation? The pellet containing the nucleic acid is washed and resuspended in a specific buffer. Then there is differential centrifugation. So in differential centrifugation, what happens? You spin it at different RPM for different duration. Then what happens? Initially the cell debris and everything comes. As you keep on increasing the speed of the centrifuge and you keep on increasing the duration of the centrifuge, the smaller molecular weight products come. Okay, the smaller size products. So see, using differential centrifugation to separate nucleic acid from cellular debris based on difference in size and density. So there is difference in size and there is difference in density, right? Centrifugation at varying speeds results based on differences. Okay, results in the formation of distinct layer containing different components of the sample. So there are different layers formed. Then what happens? Alkaline lysis. This is mainly used for the isolation of plasmids. I'm sure you remember plasmids. We have studied about plasmids in the last few classes. So we will look into this in a lot more details. In our lab, we usually isolate plasmids from bacteria. Okay. So a method commonly used for plasmid DNA extraction involving the addition of an alkaline solution to lyse the cells and denatured proteins, nucleic acids are then neutralized and precipitated using ethanol or isopropanol. So we will be looking at this in greater detail soon. Enzymatic digestion. This utilizes enzymes such as proteinase K. Proteinase K is an enzyme which goes and cleaves proteins. So this is how you separate the nucleic acid from the contaminating impurities, right? Be it protein, be it lipids, be it polysaccharides, you use enzymes to remove them. Again, if you want only DNA, then you add RNAs so that RNA gets chopped off. If you want RNA, you will add DNAs into the solution, right? So enzymatic digestion is often combined with other purification methods to improve yield and purity. Okay. Now we will look at the different methods of DNA isolation in details. So there is cesium chloride density gradient centrifugation. Now this is also called isopycnic centrifugation. So if this reminds you of picnic, that is good. So what happens in isopycnic centrifugation? So here what happens is this method is based on a phenomena of buoyant and specific density. Okay, so cesium chloride. Cesium, you know, if you remember the periodic table, cesium is very, very heavy, right? So it is a very heavy metal and chloride is a very tiny anion, right? So cesium chloride, it is having very high density. So this is an extremely dense salt, and when solutions of this salt are subjected to centrifugation at very high speed, typically 100,000 rpm, CSCl sets up a concentration gradient. So it is highly concentrated at one end and it is very low concentration is there at another end, right? So or of the centrifuge tube, and that's in, and much less concentrated at the other end. Okay, so there is basically a gradient of concentration. Then what happens if the centrifugation process is ended gently with the centrifuge slowing down slowly, and the denser solution with higher CSCl concentration will settle to the bottom of the tube, maintaining the concentration gradient for some time. Other molecules dissolved in the solution will separate themselves according to how dense they are, with the denser molecules going towards the bottom and the less dense molecule going towards the top. So did you understand? Isopycnic. Isopycnic is basically same density. Isopycnic here means same density. So what happens when cesium chloride is spun at a very high speed, then what happens? Cesium chloride gradients itself. Okay, so at the region where there is too much cesium chloride, it is more dense and it is usually at the bottom. Then as you go higher, the density of the solution decreases and therefore the amount of cesium chloride there also decreases. Therefore what happens, initially if you look at the figure, initially there was cell extract which was having DNA, RNA, proteins, lipids, everything, right? After that, the heaviest ones settle at the bottom and the lightest ones settle at the top. This is what happens. This is isopycnic centrifugation. Okay, so right now what happens when DNA is in the presence of a molecule is going towards one second, yeah. So when DNA is in the presence of a molecule called ethidium bromide, it will bind to it and become fluorescent under UV light. Ethidium bromide also adjusts the density of DNA so that it moves towards the center of the tube upon centrifugation. So contaminants will move to different positions. So you can see here in this diagram that the different molecules along with based on their density, they are segregated into different regions. Okay, and therefore they will be separated from the DNA, which can be easily seen and recovered since it will be fluorescent. So how will you know that it is DNA? You add in ETBR. ETBR, you know that it goes and intercalates between this A and T of the DNA. It intercalates between the bases of DNA and therefore it will be glowing, right? in UV light. You can see it in UV light. Then what will you do is simply separate that from the other components, which will be colorless, right? Subsequent steps will be to separate the DNA on the cesium chloride. Okay, cesium chloride, and this technique is particularly effective for separating plasmids, which are small DNA strands. If you look at the figure, linear DNA and supercoiled DNA are coming separately. Okay, and similarly for bacteria, it will be circular DNA and supercoiled DNA. Supercoiled DNA is basically the plasmid DNA. So what happens in supercoiled DNA? It is circular, it is closed, circular, and covalently bonded, right? So that is CCC, covalently closed and circular. That is basically your supercoiled DNA. Now what happens? Okay, so DNA molecules that bind to the characteristic amount of ethidium bromide, they will be seen, and then this technique is particularly effective for separating plasmids, which are small DNA strands that bacteria use to share information on antibiotic resistance with each other, and mitochondrial DNA and other specific DNA molecules that bind to characteristic amount of ethidium bromide and therefore will have a unique density. Okay, it is easy to separate plasmid DNA and mitochondrial DNA. These nucleic acids are arranged in highly coiled, once again supercoiled and circular pattern. DNA that is not supercoiled, much longer chromosomal DNA species bind to different amounts of ethidium bromide and have a different density, making them easy to separate from plasmid DNA. So using this method, you can separate plasmid DNA from genomic DNA because they are going to come at different positions in the in the tube, right? So next we will look at another technique, which is GITC and CTAB based extraction. So GITC is guanidinium isothiocyanate. So what is this? This is basically, if you remember, guanidinium is like urea. Okay, this is a denaturing agent. So what happens in guanidinium? If you remember the structure of arginine, arginine was having these 3 nitrogen in the end of its ring, right? So 3 nitrogen, and then that was called the guanido group. So this guanido group and then isothiocyanate. So that means there is a cyanide group, right? And it is basically present, and this is going to participate in separation. It is going to participate in denaturation of the proteins. When you add guanidium chloride, guanidium hydrochloride, it also denatures proteins. Okay. Now see, an aqueous solution of this salt guanidium thiocyanate is mixed with solvents, phenol and chloroform. Remember, phenol and chloroform is used for organic extraction. So GITC is added along with that to specifically separate RNA. Okay, allows for effective purification of RNA through a minimal number of steps. So when samples are grouped up and mixed up with chloroform, phenol, sodium acetate, guanidium isothiocyanate are subjected to centrifugation. The salt solution will separate from the solvents, providing an upper aqueous layer. So if you look at this, there will be an upper aqueous layer. If you can see in the figure, we will have a look at the figure to see. See, this is basically the media. From there you have got this homogenized cell lysate. It is having RNA, DNA, protein, everything. It is having guanidium, and you've added guanidium to it, and then you add phenol to it, and then there is a phase separation. So upper layer is having your what? RNA, and the lower layer is having proteins and DNA. Okay, so this upper layer, it is basically soluble. It is having this RNA, and then when you add isopropanol, what happens is it basically forms this pellet, right? So RNA undergoes precipitation, and then you remove this solution and you redissolve this precipitate with water or a suitable buffer. Okay, this is the entire story of GITC along with phenol, chloroform and isopropanol for isolation of RNA. Then you have CTBR. CTAB. So what is CTAB? This is cetyl trimethyl ammonium bromide. This is basically a positively charged detergent. Okay, so it is positively charged. This technique is most widely used for plant samples. Now remember, plant samples, if you remember, in our body, DNA is basically diploid, right? So we have two end DNA, but plants, they have polyploid DNA. Okay, so for example, wheat is having maybe hexaploid or octaploid, like that. Okay, so there is a lot of DNA in plants, a lot. So if you want to separate that genomic DNA, then you will have to use some kind of um, CTBR, which is basically positively charged, and it is used. See, so it is used for many food samples, nucleic acid and some polysaccharides. So for plant DNA separation, the major hurdle is the presence of a large amount of polysaccharides. So all these lignin, cellulose, all of this are present on the plant cells, right? And so it is very difficult to separate these from nucleic acid, and therefore 2% CTAB is used at a very high pH. So neutral polysaccharides and proteins are obtained. Okay, so neutral polysaccharides and proteins are soluble, and that is why when you give high pH, this becomes what happens, this provides an easy way to remove many difficult plant-based contaminants prior to further purification. So if you look at this method, this is basically separation of plant DNA using CTAB. So this is the plant sample. It is first grounded. Always remember, if this is taken from a natural source, it is always, first thing is grinded, and then what happens, CTAB is added, 65 degrees centigrade, that is centrifuged, and then you can see two different layers are present. So two layers are there, and then you add chloroform plus isoamyl alcohol in the ratio 24:1. So chloroform and isoamyl alcohol is added. Then what happens? Then it is chelated on ice, it is centrifuged, and then you add ethanol. Ethanol precipitates the DNA. You can see the DNA basically gets precipitated. So what is chloroform and isoamyl alcohol doing? This is basically, what it is doing is, it is assisting in this phase separation. Okay, so it is assisting in the phase separation, and all the proteins and all these polysaccharides, they are present in a separate phase, and in this green phase your like DNA will be there, and then you add ethanol. Then after that, once again you add ethanol and centrifuge. You basically resuspend this, add ethanol, and then centrifuge, and pure DNA is obtained, and then you resuspend using a specific buffer. Okay, this is the process of isolation and purification of nucleic acids from bacteria. So we will look at this. So I am sure you have understood this entire process, so you can have a look at it. So basically for your exams, what is important is exactly what is the role of each component. For example, it is CTAB. What is the role of CTAB? If it is let's say chloroform, what is exactly the role of chloroform? What is exactly the role of isoamyl alcohol? So these you need to find out, these you need to study. Specific role of each and every component. So why are we using chloroform with phenol? Okay, that is very important. So that you need to look up, and then you need to learn about those. Fine. Now we will look at the isolation and purification of nucleic acids from bacteria. So I hope you remember that inside the bacteria this plasmid was present. Inside the plasmid you have made changes, you have done this genetic engineering thing, and then you have made changes in the DNA, right, inside the plasmid. Plasmid is containing gene for antibiotic resistance. It is containing the gene for maybe a specific particular protein expression, or it is having gene for, let's say, anything. Okay, it is a modified DNA. Now you want to extract that modified DNA from bacteria. That DNA you want to go for PCR, you want to amplify that DNA, you want to send it for sequencing, you want to go for cloning with it, you want to go for more genetic engineering. Okay, so whatever you want to do, for that you need to first isolate the DNA from bacteria. So how do we do that? So see, step 1 is transformed E. coli DH5 alpha cells, hopefully harboring your plasmid of interest, were plated on agar plates. So these are basically plates. Bacterial culture is there. It's a solid media on which tiny colonies have grown. Now you scrape the colony and you inoculate that in a liquid media. Okay, so this is DH5 alpha cell. That means cloning has been done in this cell strain, right? So let's see. It has been supplemented. So inside that media, antibiotic has been added. So only the bacteria carrying this plasmid, which is again carrying gene for antibiotic resistance, only that is going to grow. Others will not grow here. Okay. So plates were incubated overnight at 37 degrees centigrade, and then you can see these small colonies, white circles highlighted by box, was picked. Okay, and placed in a tube containing 5 ml liquid LB media supplemented with 50 gram kanamycin. So this is your liquid media. This, if you can see, this is basically the liquid media, and inside the liquid media you have again added kanamycin. So only the bacteria that are having this particular plasmid in them, they are going to grow. Others are not going to grow. Fine. So tubes containing your cell suspension were incubated overnight at 37 degrees centigrade. This steps allows for the propagation of a large number of cells containing your plasmid of interest, and these cells are theoretically genetically identical as they are cultured from a single colony. So it is believed that a single colony is formed by a single cell. Okay, that is called the colony forming unit. That particular cell is called colony forming unit. So one cell divides and it forms a huge group of cells which we can see with our naked eyes. That is called a colony. Okay, and then what happens? You centrifuge it. Okay, when you centrifuge it, so prior to alkaline lysis, which is again another method for plasmid isolation, you centrifuge the whole thing. When you centrifuge, all the cells go to the pellet. So pellet is containing all the cell, and all the media or the liquid media is there in the supernatant. You throw the supernatant. Okay, you collect the pellet, and then you add the buffers gradually. We will learn about the different buffers. So this is called harvesting the cell, okay, to remove excess media from the system. This is done by centrifugation, which allows for the separation of the liquid media, that is supernatant, from the cells as a pellet. Okay. Now bacterial cells are very tough and do not typically break open by centrifugation. Fine. Then we will have to see how do we exactly break open the cells. Okay. So we will look into that. Now we will look at another method of separation, which is the phenol chloroform based extraction. So if you can see the figure, what exactly happens in phenol chloroform based extraction. See, this is the cell extract. In the cell extract, you add phenol. To the cell extract, you add phenol. Then what happens? All the different components are present. Phenol is going to act on your protein. It is going to act on the polysaccharides and everything. Okay. Then for organic method, phenol chloroform extraction using SDS, proteinase K and RNase A for the enzymatic digestion of proteins, RNA and non-nucleic acid cellular components respectively. A mixture of phenol chloroform and isoamyl alcohol in the ratio 25, 24 and 1 is then added to promote the partitioning of lipids and cellular debris into the organic phase. So leaving isolated DNA in the aqueous phase. So if you look here, this is the interface. This is the region between the two different separated phases. So in the aqueous phase, there is nucleic acid. In the organic phase, there is a huge clump of all the polysaccharides, proteins. Okay, phenol goes there. And all these are here. Now, you have the proteins at the interface, and the organic layer, all the cell debris, everything, it goes to the down layer. Fine. Then in this extraction method, isoamyl alcohol is basically an anti-foaming agent. So it prevents frothing and helps in reducing foaming between the interface and prevents the emulsification of a solution. So from here you've understood the role of isoamyl alcohol. What is the role of chloroform? This is basically pulling the phenol down. That is its role. Okay, it is basically assisting in separation of the two layers. Fine. Right. So you can see two different layers here. Now we will look at the entire process of alkaline lysis for extraction of plasmid DNA. Okay. So let us have a look at this. Now what happens here is alkaline lysis. 1 second. No, that's fine. Okay. So plasmid is an extracellular, extrachromosomal DNA. So if you remember how plasmids look like, this is your plasmid and this is the bacterial chromosome. Okay. So plasmids are very, very small compared to bacterial chromosome. They are this circular and covalently coiled. Okay, they are super coiled. Therefore, they are very different from the bacterial genome. So they are capable of independent replication. The term plasmid was first used in 1952 by Liederberg. And plasmids are very small in size. So they are around 1 to 1000 kilobase pairs. Okay. So plasmids are circular double-stranded and they vary in different bacterial species. So what happens in this procedure is that the pellet bacteria by centrifugation. First what you do is you have the media. Let's say in this entire flask a lot of bacterial cells were there. Then what you do, you centrifuge that and you get this pellet. Then you suspend that pellet in a particular solution. That solution is then called lysis buffer. So you add lysis buffer. Lysis buffer is basically lysing the cell membrane and then it is taking out all the different components out. Then you are adding the neutralizing buffer. So in neutralizing buffer what happens? Centrifuge the pellet to precipitate the protein. So all the protein gets precipitated. So what is soluble? In the soluble layer you have the nucleic acids. Then you transfer that to a spin column. As I told you, these silica based spin columns are there. You transfer the entire liquid component, the supernatant, to the spin column and then you wash and elute the plasmid DNA. Now it sounds very simple, but there are a lot of different solutions and you have to know the role of each and every solution and you have to know the composition of each and every solution. Okay, so let us have a look at this. Here is again a little bit of theory and then we will look at the different solutions used for plasmid DNA isolation. So alkaline lysis, so this tells you there is some alkali which is present. Okay, and lysis means it is basically breaking open the cell. Alkaline lysis in combination with detergent SDS. SDS is the detergent, right? Sodium dodecyl sulfate. This has been used for more than 20 years to isolate plasmid DNA from E. coli. So exposure of bacterial suspensions to strongly anionic detergent at high pH opens up the cell wall, denatures bacterial chromosomal DNA and proteins and releases the plasmid DNA into supernatant. Although the alkaline solution completely disrupts base pairing and the strands of the closed circular DNA, this is the plasmid DNA, are unable to separate from each other because they are topologically intertwined. As long as the intensity and duration of exposure of OH- is not too great, the two strands of the plasmid DNA fall once again into register when the pH is returned to neutral. So during lysis, the bacterial proteins, broken cell wall and denatured chromosomal DNA becomes enmeshed in a large complex that are coated with dodecyl sulfate. These complexes are efficiently precipitated from the solution when the sodium ions are added and they are replaced by potassium ions. After denatured material has been removed by centrifugation, the native plasmid DNA can be recovered from the supernatant. So this is the entire story of your alkaline lysis. So this is something that you have to remember. Okay, this is the entire story of alkaline lysis, and this is something that you have to remember. Okay. Right. So I hope you've understood what happened here. So basically we are adding detergent, sodium dodecyl sulfate, and we are adding NaOH. Both of these things, what they do is they are going to completely denature all DNA, all proteins, and then what happens, since the bacterial chromosomal DNA is huge, and when the two strands get separated, they are far from one another, and then what happens, we are adding this neutralizing buffer. So basically the pH is returned to normal. Then what happens, the plasmid DNA, see, it was very closely intertwined, it was covalently closed, and it is very close to one another. The two strands, even after separation, they are close to one another. So it is easier for the strands of the plasmid DNA to find one another and regain the double-stranded nature. For the bacterial DNA, however, regaining the double-stranded nature is very difficult because we are not letting it sit like that for a long duration, right? And that is why what happens, it basically gets joined with the proteins, with the lipids, with the SDS, and there is a huge complex which basically precipitates out, and then it remains at the bottom of the tube, and you get the entire uh, plasmid DNA in the supernatant, and that is basically separated out. So that is the entire story of your alkaline lysis. Okay, so there is denaturation followed by renaturation, but very selective for only the plasmid DNA. The genomic DNA remains denatured, and it is all intertwined with the cellular proteins. So I hope you understood this. Okay. Now we will look at the different solutions used. Now this is very interesting. You need to know the different solutions that are used in this case. Okay. So after the entire bacterial culture has been pelleted down, that is, the cells have been harvested, all the media has been thrown, then what happens? You are adding tris buffer. You are adding EDTA. What is tris doing? It is giving a specific pH. It is giving a pH of around what, 8 or something, right? Or even higher pH. Then there is EDTA. EDTA is going to chelate the divalent cations. Now DNases need divalent cation for their activity. What is the activity of DNase? DNase is going to cleave DNA. We do not want that, and that is why we are adding EDTA, so that it can catch hold of all the cations, divalent cations, and make sure DNA is intact. It is not chopped off by the DNases. Fine. Then what is there? Glucose is added. Glucose gives a osmotic balance. Okay. Then there is RNase A. RNase A degrades the RNA once the cells are lysed, because this is for plasmid DNA isolation. It is not for RNA isolation, and therefore we want all the RNA to get removed. Fine. Then there is lysis buffer. That is basically your resuspension buffer or solution 1. What happens in solution 2? So solution 2 buffer contains NaOH. Here the alkaline lysis basically begins. So there is NaOH. NaOH is going to break down the cell wall, disrupt the hydrogen bonds between the DNA bases. It is basically going to denature the DNA. This results in DNA denaturation into single strands of DNA. SDS is there, which coats the proteins, denatures the proteins, solubilizes the cell membrane. That is the work of SDS. Fine. Then there is neutralization. So what is happening in neutralization or precipitation buffer? Here potassium acetate salts are added, the pH is changed. Tris is having a very alkaline pH, but here it is acetate pH, right? So it is basically dropping the pH and serves to decrease the alkalinity of the preparation and reform the hydrogen bonds. So hydrogen bonds are now getting reformed, but only selectively for the plasmid. For the bacterial DNA, it is impossible to get all those bonds reformed in such a small amount of time. So what happens in this step, we must consider the small size of the plasmid DNA in comparison to the chromosomal DNA. The plasmid double-stranded DNA is readily dissolved in the solution, but the large genomic DNA is tangled in a meshwork of denatured cellular proteins and other cell debris, mainly driven by hydrophobic interaction. This results in a white, fluffy precipitate. Fine. Now we will go to the next slide. So this is the third last slide for your theory. Fourth last, okay. Next few slides are not that difficult, okay. So what is trizol? Trizol is an agent which is used for isolation of RNA, right? So what is there in trizol? Trizol is basically having three components, and that is why it is called trizol, right? So what are the three components of trizol? It is having phenol, it is having chloroform, and it is having GITC. These are the three components of trizol. Now besides that, it is having a pH of 7, and it is having also other salts like sodium citrate, lauryl sarcosine, and mercaptoethanol. This is basically used for the isolation of RNA. If you see, there are two different phases after addition of trizol reagent. The aqueous phase is having only RNA, interphase is having DNA, and the organic phase are having protein and lipids. So low acid, the low pH or acidic pH of trizol controls to separate RNA from DNA and protein, while a high pH can cause RNA and DNA to be isolated together. For DNA isolation, we were using high pH. Remember? Now guanidinium salt serves as a chemotropic agent to denature proteins, and phenol, indicated in pink color, is an organic compound that is used to extract nucleic acids and proteins. Excuse me. Okay. So after solubilization and homogenization of the samples in trizol, RNA, DNA and protein are differentially extracted by addition of a phase separation reagent, which is phenol chloroform. You have studied that phenol and chloroform are basically causing this entire phase separation. This is clear? Fine. The solution separates the RNA away from DNA and the proteins into different layers. Subsequently, the RNA in the upper aqueous phase is then collected by alcohol-based precipitation using isopropanol or ethanol, and then it is air dried, resuspended in the final sample buffer. Got it? So this is how RNA is isolated using this particular trizol reagent. Fine. Then we will see, now we have isolated RNA, we have isolated genomic DNA, we have isolated plasmid DNA. We have isolated all of these. Now how do we know how good our isolation process was? So we need to know two things: how much DNA is there and how pure is the DNA. So how much DNA or how much RNA is there, that you can know from your absorbance test. Okay, so UV spectroscopy you can do, UV visible spectroscopy, to know how much DNA or how much RNA is there, and then you can check the purity of the RNA or DNA using agarose gel. Okay, so agarose gel can tell you exactly how pure the sample is, and also you can know about the purity, you can get some idea about the purity from UV visible spectroscopy. So let us look at that. So quantification of the purified product. So this is the graph obtained after absorption. Okay, after you check absorption. So at 260 nanometers, what happens? The nucleic acids are absorbing. So nucleic acids are absorbing UV visible light, the adenine, thymine. So all of these, 1 second. Yeah. So the nucleic acids, adenine, thymine, uracil, guanine, cytosine, these are having, sorry. Okay. So yeah, so now these are having these alternate double and uh, single bonds called conjugate double bonds. So because of this structure, they are basically absorbing light in UV visible range, and then it is detected, right? But then the salts in the buffer, for example, there is phenol, and then there is phenol and different salts, they are absorbing at 230 nanometer. Okay, protein backbone also absorbs at 230 nanometer. Then there is aromatic amino acids in the proteins, which are basically absorbing at 280 nanometer. So there is tryptophan, there is phenylalanine, and there is tyrosine, right? So these are absorbing at 280 nanometer. Therefore you check the ratio, you check the ratio of 260 with 280 and 230. So if your ratio for pure DNA 260 by 280 ratio, it should be higher or equal to 1.8, and for pure RNA the 260 by 280 ratio should be 2, and the 260 by 230 ratio, this should be 1.8. So that is ideally what should happen. Okay, but if there is more phenol, then what happens? You can see a peak shift. The peak is going to shift towards this 230 nanometer. If there is more of protein, the peak is going to shift towards the right. So this will be telling you about the purity of your samples as well. Fine. So then how do we know about the concentration of our samples? For that, what we need to do is, see, this is the formula for concentration of DNA sample in microgram per ml is basically equals to your dilution factor into absorbance. So what is this 50? Right. 1 second. So you can see here a ratio of 1.8 is generally accepted as pure for DNA, and a ratio of 2 is generally accepted as pure for RNA. But then why is that so? Because amongst the 5 nucleotides, if you see, uracil has a very high absorbance. Okay, uracil's absorbance is 4, adenine's absorbance is 4.5. And since thymine is replaced by uracil in RNA, therefore the absorbance of RNA is much more compared to the absorbance of DNA. Besides that, RNA is having the bases, they are more exposed to the environment, and therefore again more absorbance, right? And that is why RNA will always give you more absorbance compared to DNA. Fine. So from this formula you can find out the concentration. So concentration can be obtained. You can check the molar extinction coefficient for DNA. How much is the molar extinction coefficient, that you can find out. Fine. Right. So then we will see the once again quantification. Yeah. So this is the one for salt, and here you will get the yield for DNA and RNA. Here you will get the yield for protein. So pure RNA will give you an yield of 1.8 to 2. If it is less than that, that will tell you that there is more of buffer contamination, right? This method does not discriminate between RNA and DNA. It is advisable to treat the RNA sample with RNase free DNAs to remove contaminating DNA. Again, 260 by 280 ratio to 260 by 230 ratio should be above 2. A low 260 by 230 indicates presence of cheotropic salts, phenols, proteins due to insufficient washing. Okay, so I am sure that is clear. Now let us start with the questions. So this is the first question. Please answer in the chat box, and we can then discuss the different answers. Start answering in the chat box. So the first question is, RNA is chemically more stable compared to DNA, and that is because. So we learnt about this in today's lecture. You have to go back to the structure of RNA, and then you can give me the answer. So the first option is presence of hydroxyl group at the third position. Second is presence of hydroxyl group at the second. Presence of ester bond at third, and uracil, RNA has uracil while DNA has thymine. Well, your right answer should be the presence of hydroxyl group in the second position. So this is ribose sugar and this is the RNA. Okay, but for DNA it is deoxyribose. Therefore, this OH group is not there. So as we learned in a few slides back that there is a nucleophilic attack by OH in an alkaline solution, and that is why what happens, this uh, OH now attacks this particular phosphate which is joined to the 3' carbon, and then what happens, the bond between this P group, bond between the phosphate and another nucleotide breaks, and there is a 2,3 cyclic monophosphate intermediate derivative, that what happens, that undergoes cleavage, hydrolysis, and then we are left with a broken RNA chain, and that makes RNA less stable compared to DNA. Okay, now see, this explains the theory behind this entire image. So RNA is made up of ribose carbohydrates rather than deoxyribose. It is more unstable than DNA because of the presence of hydroxyl group, which renders it susceptible to hydrolysis and destruction. In general, RNA is unstable due to its chemistry. So the absorbance of this hydroxy group in the DNA, also known as 2' deoxyribonucleic acid, is significant because RNA has a typical 2' hydroxyl group. The phosphodiester bond is unstable and prone to nucleophilic assault and self-hydrolysis. Right, so that is the entire explanation. Now we will look at the next question. So next question is also very easy. They are asking you what is the reason for the isolation of DNA from natural sources being so challenging. So the right answer would be which one? High amount of RNA is present, high amount of protein is present. Okay, so you are saying it's C. Yes, that is very correct. Now see, in this case from plant sources you might be tempted to go for low amount of DNA. But low amount of DNA, as I told you previously, this is not going to be correct because for plants exist as polyploid, right? So the DNA present in plants, it is tetraploid, it is hexaploid, right? Octaploid. For example, wheat. If you go and check, wheat is actually, I think, hexaploid, like that. So plants, they are having polyploid DNA, and that is why DNA is present in very high amount in plants. But the issue with the DNA is that there is a lot of secondary metabolites, a lot of polysaccharides such as cellulose, lignin, all the secondary cell wall is there, right? Primary, secondary cell wall, and it is very difficult to separate all these polysaccharides from the actual DNA, and that is why it is very difficult from natural sources. Fine. So you can have a look at this. See the cell wall composition. It is made up of cellulose, hemicellulose, lignin. Okay, breaking down these barriers to access the DNA within the cells can require robust mechanical and enzymatic methods. Then again, your secondary metabolites like phenolics, polysaccharides, polyphenols, these can interfere with the DNA isolation procedure. Okay, then again nucleases and polyphenols and polysaccharides, this is again there, this can bind to DNA forming complexes that hinder its isolation, and therefore it is very important that we remove those compounds, and to remove those compounds we are going to use this C-tab isolation method. C-tab is carboxy, what was the name for C-tab? You can go back to our previous slide and go through this. We have already discussed cetyl trimethyl ammonium bromide, right? This was the method for using C-tab we can isolate DNA. So we are going to do that. Fine. Then we have the next question. So next question is again simple. You have already learnt about this in our previous slides. This is asking you what is the function of EDTA in cell lysis. So you tell me acts as a metal chelating agent, which is definitely correct. Denaturing protein, destabilizing cell wall, and all of the above. Okay, so let us look at the question. Now let us look at the answer. Okay, right answer is A. This is the structure of your EDTA, and using all of these groups, it is going to chelate the metal ions. See, so divalent cations such as Mg2 plus Ca2 plus, these are required for DNA's activity to break down DNA, right? And therefore EDTA chelates the divalent cations in solution and prevents the DNAs from damaging the plasmid. Therefore it is also helping in destabilization of the cell wall. Okay, so it is actually doing both of these things, but the more appropriate answer would be acting as a metal chelating agent. Fine. Then we will go to the next question. Okay, so there are two different questions. Try to answer these two questions. What is the function of NaOH in lysis buffer? It is again a simple question. What is the role of SDS in lysis buffer for plasmid DNA isolation? Okay, let's see. So NaOH, A and C. Okay. So what is the function of, okay, so that's an MSQ, right? So are you answering for the first question on this page? Functions of NaOH lysis buffer. Denaturation of genomic and plasmid DNA, that is correct. Degrades the RNA. No RNA is going to degrade the RNA and breaks down the cell wall. Removes the protein from the lysate. Okay. And then what is the role of SDS in lysis buffer? Solubilizes the membrane, denatures the protein, disrupts the RNA protein complexes. So SDS is going to do all of these, and yes, you're very right. It is the right answer. Right. So you can have a look at this. So when we were studying the isolation of plasmid DNA, we looked into the work of the function of the lysis buffer. Lysis buffer is actually containing your NaOH and SDS. So NaOH is going to break down the cell wall, disrupt the hydrogen bonds between the DNA bases, right? It is going to denature the DNA, separate the two strands, and then SDS is going to destabilize the membrane. It is going to denature the cellular proteins. So yes, that kind of explains the functions of both SDS and NaOH, right? Then there is one more tiny question. It is again an MSQ. Tell me, isolating plasmid DNA from genomic DNA is based on. So we have studied about this isolation of plasmid DNA, and we know that plasmid DNA is isolated from genomic DNA using a specific principle of plasmid DNA, which is a characteristic of the plasmid DNA, and that differentiates it from the genomic DNA. So what is that? Okay, so you're saying it's conformation and it is size difference. Yes, that is very correct. So conformation is, it is circularly coiled, covalent, and it is very small. So small tells you about the size difference, and conformation is circularly coiled and it is covalent, and therefore what happens during the neutralization reaction, it is easier for the strands of the plasmid DNA to quickly rejoin, to actually form these hydrogen bonds, right? So they are in each other's vicinity because they are not separated very far. So if you look at this neutralization buffer, the buffer contains potassium acetate, pH is given, serves to decrease the alkalinity of the preparation and reform the hydrogen bonds between the complementary single-stranded DNA bases. In this step, we must consider the small size of the plasmid, right, DNA in comparison to the chromosomal DNA. The plasmid single-stranded DNA, double-stranded DNA is readily dissolved in solution, but large genomic DNA remains tangled up with all the denatured cellular protein, all the cellular debris, right? So that results in a white fluffy precipitate. Now we will look at the next question. So this tells you about the conformations of different plasmids. Okay, so as we were talking about the conformation of plasmids here, I have included this slide for you so that this gives you a better idea of the conformation of plasmids. So when you run the plasmids on a agarose gel, you can see that the different conformations of plasmids can be distinguished because they run differently. Okay, so nicked open circular. So this is open circular and only one strand is broken. Okay, it has one strand cut. Relaxed circular, it is basically intact and the supercoils are removed. Linear, it has free ends. Okay, then there is supercoil. This is your plasmid DNA. So covalently closed and circular. DNA is intact, both the strands are uncut and with a twist built in, resulting in a compact form, and that is why the supercoiled DNA is running faster than the nicked DNA. Then there is supercoiled denatured DNA. Supercoiled denatured DNA is also going to run faster than nicked DNA. Okay, so the conformations are listed here from slowest to fastest, and for a given reason they run at different speeds in the gel electrophoresis. Fine, then we will go to the next question. So what is the composition of your elution buffer, and when the neutralizing buffer is added after lysis to the cell lysate, then what do we see? Okay, so elution buffer contains tris. Fine, elution buffer does contain tris, and elution buffer, okay, then when neutralizing buffer is added after lysis to the cell lysate, then what happens? Chromosomal DNA gets tangled and precipitates out, that is true, and then plasmid DNA anneals and remains in solution, that is also true. Proteins and SDS form insoluble precipitate, that is true. Therefore all of the above should be the right answer, right? Let's have a look. Yes, so elution buffer contains these two, tris and EDTA, and the neutralizing buffer contains all of the above. Fine. So why does elution buffer contain EDTA? To inactivate the DNases, because this is isolation of DNA, right? Plasmid DNA isolation, and therefore we don't want the DNases to be active. The DNases should not be chopping up the DNA. Fine. Now during extraction of plasmid DNA, after addition of neutralizing buffer while precipitate, white precipitate is formed due to insoluble potassium dodecyl sulfate salt, soluble potassium dodecyl sulfate, soluble potassium acetate. Now always remember, precipitate is formed when something is insoluble. Otherwise it would be a clear solution, it would be intertwined with the entire buffer, right? So only and only when it is insoluble it is going to precipitate out, and therefore yes, A should be the right answer. Fine. So you can have a look at this. This is how the entire fluffy white precipitate looks. So the large genomic DNA along with proteins, along with SDS, all of that cell debris together form these white fluffy precipitates, and here your supernatant is present, and in the supernatant the entire plasmid DNA remains. Okay, and this precipitate is formed because of insoluble potassium dodecyl sulfate instead of sodium. Right, so SDS is definitely soluble, but KDS is not soluble. So now we have two more questions. DNA bound to silica membrane is eluted by the addition of A and C. Okay, so this tells you, this is asking you about the spin column that we were talking about. Okay, so the DNA goes and binds to the chaotropic salt. Chaotropic salts bind to the silica. Now how are we going to remove the interaction? To free that interaction, we are going to add some kind of buffer that is going to overtake, right? That is going to take your DNA down and it is going to bind to the chaotropic salt. Okay, now we have to wash the DNA bound to silica membrane. Which of the following reagents are used to wash the DNA bound to the silica membrane? Okay, so you are saying it's C. Fine, let's see, let's have a look. So for the first question, it should be alkaline conditions. Okay, and for the second it should be ethanol and isopropanol. So if you look at this, yes, you're very right, it is A and C. So there should be nucleus free water, and it should be low ionic salt buffer. Okay, so here it is explained that TB is supposed to be given, TE, which is the elution buffer, and then low ionic salt buffer is given. And then for the next question, we have this. What happens? Ethanol is added. Ethanol dissolves the salt but not the DNA. And so then what happens is the DNA precipitates out, but this can also interfere later. And so therefore it has to be spun properly. Ethanol needs to be removed. So low salt and higher pH, silica membrane becomes negatively charged like DNA, and so they repel. Right, and that is why low salt buffer is given. Higher pH has to be there. The silica membrane becomes negatively charged, and DNA, they are going to repel. Right, and that is why all the sodium is actually taken away, all the cheotropic salt is taken away, and therefore there is nothing to bind the DNA with the silica membrane. Therefore DNA comes out. To wash the DNA bound to silica membrane, which of the following reagents are used? It is ethanol or isopropanol. Now we have 3 more questions, and then I think we will be done after one question just. Fine. So try to answer. The quality of the purified plasmid DNA can be checked by SEC, agarose gel electrophoresis, UV spectroscopy, and SDS page. So quality, not concentration. Yes, very good. Agarose gel electrophoresis is going to tell you about the quality of the plasmid DNA. Then you have neutralization buffer. So neutralization buffer should definitely have, you know what it is, potassium acetate. Fine. Then in the agarose gel electrophoresis, the plasmid DNA runs ahead of the linear DNA because linear DNA is more compact. No, plasmids have higher mass or charge ratio. That is wrong, because plasmid is supercoiled and migrates faster. Yes, so plasmid is not going to have higher mass or charge ratio compared to the genomic DNA. Right? Okay. So you're answering for neutralization buffer, right? So that is C, correct. And then for the last question, it is C. So in this case, you can do UV spectroscopy, you can do agarose gel electrophoresis. For this question, it is C, and for again this question, C would be the right answer. Fine. Then we have one more question. It is about RNase A. So what happens if you forget to add RNase A to the cell lysate? Okay, and you are trying to isolate DNA. What is going to happen? Try to think and give the answer. So A, DNA will be degraded. That is not the case. RNAs will not be degrading DNA. RNA will not be removed, as both DNA and RNA absorb at 260 nanometer. Quantification of pure isolated DNA will be problematic. That is very true, because RNA is going to absorb more than DNA. Both DNA and RNA will form a complex and precipitate out of solution. That is not going to be the case. All of the above is not going to happen. So yes, D would be the right answer. Right, and that is why it is very important to separate RNAs and DNAs. Fine. So that is all. These are the references. You can go through them. There are references for websites, there are references for the books that you need to know for referring to this topic. Thank you so much for attending the class, and thank you for answering the questions. So if you have any questions, you can ask me, and then we can end the session. Thank you so much. Srishti, you should also be answering the questions sometimes. Okay, should we end the class then? Yes, today my net connection is good, right? Thank you so much for attending the class. We will see you in the next class.